All right. Well, welcome everybody. Thank you so much for joining us for the 2022 Teen Science Scholar Showcase. My name is Eric Shames. I am the volunteer manager here at the Denver Museum of Nature and Science, and I also help manage the Teen Science Scholar program every year. I'm extremely fortunate to do so. So some quick housekeeping for everybody. The agenda today is we will hear from two of the members of our TSS Advisory Committee to talk about the program overall, and we have a short video as well to share. And then we're going to throw it over to each of the areas of Teen Science Scholars to show off all the hard work that they have been doing this summer. We're really excited to see all of that. If you're an attendee today, thank you again for coming. But just a heads up, we can't see you or hear you in the webinar, but you can use that chat function on the bottom bar over there to type in words of encouragement, congratulations. And if you have questions, we will save those for the end. And you can go ahead and ask any questions of our panelists, of scholars, and have them answer them at the end of the session over here. So like I said, this is an absolutely amazing program that I feel extremely fortunate to be involved with year after year. It's a program that invites 20 rising juniors and seniors in high school from all across Denver and the Front Range to work alongside some of our amazing, outstanding science staff here at DMNS to work on various projects that they're working on. Obviously, this is not your typical internship program. They really get a lot of great behind the scenes and hands-on experience in various scientific fields. So like I said, it's an absolutely amazing program I love to be a part of, and it really does take a village to make the program run. So I want to quickly thank all of the mentors who are a part of this program for all of the work that you do. All of the friends and families of our scholars for supporting your scholars to be a part of this program. I want to thank the donors and contributors for making this program happen year after year. Thank you so much for your kindness and generosity. The Advisory Council, thank you so much for all of your hard work. And of course, the scholars, thank you guys for an absolutely amazing summer that it's been. It really flew by far too fast, but it's been amazing to get to know you all, and thank you for your hard work. With that being said, I would like to first introduce Dr. Tyler Leeson, who is our curator of vertebrate paleontology, but also plays a really pivotal and primary role with me in planning and figuring out all the logistics of the program to make it happen year after year. So with that being said, I'm going to throw it over to Tyler for a few words.
All right. Well, good afternoon, everyone. And thank you for attending this year's Teen Science Scholar Showcase. I'd like to second everything that Eric said. I'm very happy to be part of this amazing program. I'm a paleontologist here at the museum and part of the team that helps oversee the Teen Science Scholar program. This is an incredibly impactful program that was started by my predecessor here at the museum, the late Dr. Richard Stuckey, over 20 years ago. And the program initially only offered opportunities in paleontology, but has since expanded to all of the sciences within our division here at the museum. So like I said, like Eric had said, the teams work alongside the various museum scientists and work on whatever the scientists are working on. So this means that every year is unique. So this is truly a very special internship. There's really nothing else like it that I can think of. And I always look forward to hearing about what the teams learned and then also what they accomplished during the course of the summer. And this program has countless success stories, and you're going to hear about one shortly. And I just love hearing about these success stories. And I think they are a true testament to how impactful and awarding this program really is. And yet, while the program is awarding for both the mentors and the Teen Science Scholars, I also know the program is a lot of work. So I'd like to acknowledge and thank all of the mentors this year. Everyone did a fantastic job, and I'm looking forward to hearing from all of your Teen Science Scholars. So with that, I'll turn it over to Mieko Zanetta.
Hi, everyone. Thanks for being here today. My name is Miekka Lunetta, and I'm part of our advancement team here at DMNS. So I work in our development department, and I do our advancement department. I do development work. So again, like Eric said, we just really want to thank the donors for all of their support that they've given to this program. This program wouldn't be possible without all of the people that support it. So again, if you look at this list, I know you can't hear, we can't hear you and we can't see you, but if you want to give a little round of applause or for the people here that have made the program possible. And if you are one of the folks on this list, thank you so much, because we really couldn't do it without you. And if you're interested in learning more about how to support the program and you're not on this list, just come contact me or come contact our office, and we'll get you set up. So like Tyler said, we have a success story that we want to highlight for you today, and we really hope you enjoy it. So I'm excited to hear more from this year's Teen Scholars, and thanks again for joining us. And thanks for all of our supporters here.
I got interested in paleontology from a super young age. I feel like every kid goes through that dinosaur phase, and I feel like I kind of never grew out of it. I'm Isaiah. I've been with DMNS on and off since I was about 16 years old. I started as a teen science scholar here, went on to college where I got my degree in biology with a focus in vertebrate evolution and vertebrate behavior. And then now I'm going on to get my PhD, where I'll have my focus in evolutionary biology, studying mammal evolution and mammal taxonomy.
So my name is Tyler Leeson, and I'm a curator of vertebrate paleontology here at the Denver Museum of Nature and Science. So Isaiah is the first teen science scholar to go on to become a PhD paleontologist. We've had other teens that have gone on to get their PhDs in other fields, but the program was started by Richard Stuckey over 20 years ago. And of course, Richard, a paleontologist, and the program was started by having teens join him in the field. It was very much a field-based paleontology program. And so now to see the program kind of come full circle with Isaiah, joining me in the field, in paleontology, and then going on to get his PhD is just an amazing feeling. As part of the program, he started to learn what it was like to be a scientist, to ask questions, hypothesis-driven questions, and then learn how to be a field paleontologist. So he was able to learn the skills necessary to go out in the field and be a successful paleontologist.
This program made science a lot more tangible for me. Before this, neither one of my parents had a background in science or a college education, so going to college is something I knew I had to do. After doing this program, I knew that this is what I loved, and also it's possible to do this. So this program made it more tangible and gave me the opportunity to see that it is possible to pursue what I want to do. I think that the Teen Science Scholar program was really key for building the next step in my career. It's given me connections and introductions into the field that I couldn't have gotten without it. Specifically, having mentors like Dr. Leeson as a great resource, and Kristen giving me advice on my research and giving me resources, or even having access to Dr. Kraus, who's a world-leading expert in a field that I want to study, who constantly gives me advice and oversees my research. I think that those things are invaluable, and the fact that I could not get them anywhere else besides DMNS, and I'm truly grateful for that.
He's, I think, on to an incredible scientific journey. Again, just watching that full arc from just a tiny little bud, like with little questions, and then you can see his mind, his scientific mind getting bigger and bigger and bigger. And I'm really excited to see it continue to grow and to see what he's able to do in the field of paleontology. I know it's going to be amazing. I mean, it's important to fund this program because it's truly one of the most impactful programs we have here at the museum. The stories, like Isaiah's story, is proof of that, where we have these teens that join the program, and they're sort of budding scientists, and then they're really able to fully blossom here at the museum, really spread their wings and go on and become full-fledged scientists. And I think that's just, for me, one of the most rewarding things about this program is seeing the teens, interacting with them, of course, during the course of the program. But then I think even more importantly is hearing from the teens afterwards and hearing their success stories, writing letters of recommendation for them, and then seeing them get these scholarships and seeing them go on to PhD programs, and then even seeing them afterwards as young professionals. And I think more broadly, we need this program because we need people to understand science. They don't necessarily have to go on and get their PhDs in science. That's great, but they need to have an understanding of science, and because that's so important for so many issues facing us today. It's just a basic understanding of science. And so this program is truly integral to the museum, and I would argue to society as a whole.
Awesome. Well, as you all can see, especially the scholars, it just shows, or comes to show that this internship, even though it is coming to a close here, can really be just the beginning of the very bright futures that I know that all of you have ahead of you. So thank you to Mieko and Tyler for help putting that all together. But with that being said, it's time for the stars of the show here, and that is our teen science scholars. So we are going to start off with our scholars who had worked in our anthropology department this year. So anthro scholars, go ahead and take it away.
Hi, my name is Faria. I go to Eagle Crest High School.
Hi, my name is Sue and I'm from Sheridan High School.
My name is Eileen and I go to Abraham Lincoln High School.
Charlotte East High School.
Ethan Thornton, Regis Jesuit High School.
My name is Eric and I go to Arvada West High School.
Eli Campbell, DSST Montview.
I'm Kaya Havley and I go to East High School.
We, the Teen Science Scholars for Anthropology, were assigned to filter through excavations from two sites, Jones Miller and WS Ranch. Flotation. Throughout these 2 months, we have gained important knowledge of ancient native groups that lived at these sites thousands of years ago, while also learning vital scientific skills and methods along the way.
Every day, we come in and set up our floating stations, as well as sorting the samples from previous days that were already floated. This process, which is known as flotation, is a scientific tool used to sort through soil samples with water and separate them into two parts, light and heavy fractions.
We usually find materials like charcoal, pollen seeds, pottery sherds, bones, and obsidian. The next day we bag up all these artifacts and organic materials with their tags, then randomize 10% to keep.
Jones Miller's is a site that dates back 10,000 years ago, and it is located in northern eastern Colorado near the Nebraska border. It was used by Paleo-Indian people for hunting bison, known as Bison antiques. Our work with this site is dedicated to learning about the people that hunted those bison and their lives and culture. By analyzing samples collected from the site, we find information on the plant life as well as some of the hunting practices of these groups.
WS Is a site that existed about 1,200 years ago, and it's located near Alma, New Mexico. It was a place in which a diversity of major native nations lived, such as the Mimbres Mogollon, the Hohokam, and the Chaco. So by observing our findings, we not only find out about their daily lives, but we figure out the political and social affairs of these ancient peoples.
Our first field trip this summer was to the Colorado Wildlife Arsenal. After weeks of working with ancient bison bones and samples from the Jones-Miller site, seeing modern bison in person really helped put things into perspective and give us further insight into how bison antiquus may have lived.
When we went to the CU Boulder Museum of Natural History, we wanted to see connections between their work and ours. Their anthropology exhibit gave us a lot of information about sites similar to Jones-Miller, including their collection of projectile points from around Colorado that were used to kill bison antiquus. In addition, we got a tour of their archives, where we were able to see some pottery made by the Mogollon and Mimbres groups that once inhabited the WS Ranch site.
We visited the Jones-Miller site in Ray, Colorado. We first went to the museum where we found projectile points and a life-size bison antiquus from the site. After that, we went to the physical site, where it now looks like a wheat field, but we imagined what it once was like when Native Americans were killing bison twice their size.
Well, dun, 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 sludgy, sludge, dun, dun.
Yeah. I threw some dirt in the tub.
Don't ask me, I'll never scrub.
Watch it turn into mud.
But now it's in my way.
I'll trade my day for a find.
Artifacts all in my mind.
I wasn't very inclined.
But now it's in my way.
Ripped bags, sample showing.
Hot day, wind was blowing.
Where you think you going, baby?
Hey, I just met you, and this is crazy. But here's my sample, so float me, baby. It's hard to read right. Ask you, baby. And here's my sample, so float me, baby.
Points right there makes it really stand out. It's one of the best pieces in this museum.
Are actually a metaphor for like life.
Oh wait, guys, we're just staring at the standing block. There's even nothing in there.
Damn.
Yeah, but it's captured for posterity.
Awesome job, Anthro team. I need to figure out how to get that song as my new ringtone. So if you can send me the clip, that'd be great. Faria, do you want to say anything?
Yeah. I hope you guys enjoyed that. We spent a lot of time on the video, especially the song as well, during our 5-hour field trip. But yeah, it was a very fun experience for anthropology. And even though we didn't do a lot of like microscopic analysis or a lot of research, I feel like even like the aspect of just doing flotation and then going out to see the sites and all of those kinds of things, it was very enriching to see, and it was honestly a great experience for teaching all of us anthropology scholars about what anthropology truly is, and finding out about ancient groups of people all the way 10,000 years ago and 1,200 years ago. So I hope you guys enjoy that, and if you have any questions, just ask in the Q&A.
Thank you, Faria. Thank you, Anthropology. Awesome job. Great video. All right, so up next, we are going to throw it over to our scholars who worked in conservation this year. Jacob and Ellie, take it away.
Nope. This one?
Nope.
Disappeared, which is super painful.
There it is. Okay.
Hello, my name is Jacob Ramos, and this is my partner,
Ellie Vanas, and we were the two teen science scholars working in the conservation lab this summer. Of our two major projects, the first we're talking about is Conifer Tones, which were excavated about 10 years ago from the Ziegler Research Site up by Snowmass, Colorado, which is up by Aspen, Colorado. What we were doing was taking these cones, modern cones, because we didn't want to destroy or hurt the ancient ones that were degraded down to the level of the ancient waterlogged cones by boiling them, putting them under vacuum and freezing them, and putting them in 3 different solutions to try and preserve them. Because waterlogging means they're preserved in water, but it also means it breaks down the cellulose and cell walls of the actual cones. So the 3 solutions, sorry, were polyethylene glycol or PEG at 2 different molecular weights, 400 and 1500, and chitosan, which is ground up shellfish shells. Both of these solutions were really working to go into the conifer cones, into the cells really, and make a sort of barrier to make sure that these cells don't collapse after the cones are dried. They don't shrink, they don't crack, there's no distortion. And then our drying process is to put them in a freeze dryer in order to try and make sure there isn't any, it dries thoroughly. These cones, the ancient ones that is, come from about between 130,000 years old and 70,000 years old. And you see on this slide, actually, the top picture is an overhead view of the Ziegler Research Site. The one in the corner is a diagram of the site over the years, how it might have changed, how it changed and all of that. And then the picture on the left is a current diagram of the different layers and stratigraphy of the site. And then here you have we have pictures of us actually making the solutions that we put the cones into. Some of the cones in storage before we did any experimenting on them. Some of the cones right after we put them into solution and after a little time had passed, and cones and scales after we had dried. They had been in the freeze dryer. And of course, us putting the solution into its compartments.
The second major project we worked on this summer was the Northwest Coast Grant, which is funded by the Institute of Museum and Library Services. What is the Northwest Coast Grant? The Northwest Coast Grant is a grant that allows DMS conservators to study and help stabilize artifacts from the source communities along the Northwest coast from Washington to Alaska. The communities involved in this grant are the Munchanuth, Haida, Miekka, Clinket, and Kwakiwak communities. 2 ways we personally contributed to help in the stabilizing and studying of the artifacts from the grant were by helping to stabilize a Clinket cedar bark mat while also studying and creating condition reports on artifacts from the Northwest Coast Grant as well. In the corner, there is a picture of right here. Right here is a picture of recorded. Here is a picture of the source communities along the Northwest coast. And over here are some of the items we did condition reports on. Condition reports. What are they? Condition reports are reports that document the current state of an artifact before treatment / course of action. How is this done? A condition report looks for what the artifact is made from, how it might have been made, what the shape and color of the artifact is, what it might have looked like when new, are there signs of use on the artifact, is the artifact missing anything, and what might be the most fragile part of the artifact. Why is this important? Condition reports are important because they help provide information to future conservators on how best to stabilize and store an artifact in its current condition. Right here are two pictures of artifacts that both me and Ellie have done condition reports on. These are some more pictures in detail of areas of cracks, losses, and of areas of damage as well.
The final section of our work this summer was on the Clinket cedar bark mat, which was made around the 20th century and found in Angoon, Alaska. It's about 6 foot by 5 foot in dimensions, and it's a mat that was used as a general floor covering, like a rug, woven out of cedar bark, some sections of which were dyed with plant-based dyes, probably. What we were doing is stabilizing it under the advice of Clinket source community members, which is what this top picture is of, those Clinket representatives talking with our main conservators, Catherine and Megan, about how best to handle it. What me and Jacob were doing, we're making bridges out of paper, Japanese tissue paper, which had been painted to match the color of the mat, cut into small strips and glued down over breaks in certain strands of the cedar bark using wheat starch paste, which is just wheat starch and water mixed together, which makes a strong but flexible bond that can be reversed with water in case we make a mistake or a conservator in the future wants to do something different, undo it, that sort of thing. This bottom picture here is really just to give an idea of what the mat looks like. And here we have a picture of the full mat, the two completely disconnected segments of the mat that came off entirely. And then us actually working on the mat. Here is Jacob vacuuming and me vacuuming very lightly, very gently to make sure we don't break anything. And then us actually working on making our bridges and working to stabilize it so it can continue to be enjoyed and displayed or learned from, really just exist for years to come. That's all, folks. Thanks for coming and listening to us.
Awesome job, Ellie and Jacob. Thank you so much for sharing all the hard work that you did, some really delicate work that you guys did over there. So fantastic job. Thank you for sharing. All right, so next we have our geology scholars. Wesley, take it away, my friend.
Is James in here?
I'm geologically right on time.
There you are.
He is.
Howdy, everybody. Wes, do you want to share your screen?
Yep. Here you go.
Perfect. So my name's James Hagedorn. I'm a geologist at the Denver Museum, and one of the projects that we worked on over the summer with Allie and Wes was trying to figure out how to visualize what the Rocky Mountain area looked like in deep time. And what you're seeing in the background of the slide here, Wes is just going to outline the boundary of Utah right now, just so you get a sense. But this is a image of what the Rocky Mountain area looked like hundreds of millions of years ago. And you'll see, for example, that northern Wyoming was underwater at that moment in time, as was parts of Idaho and northwestern Utah. And so one of the things that we worked on this summer was trying to figure out how to use fossil data as well as data from rocks to create these types of maps. And this isn't the map that we worked on, but Wes is going to tell you how we did that and what we learned.
Cool. Thank you, James. So the first thing you want to look at is stuff on the surface, which is outcrop. So this is a map from the late Jurassic, and we're going to be using this as kind of a case study to show what we did. This is about 157 million years ago, so pretty long ago. And so what we have here is we have this map. You can see There are 12 states included in here. Some of them are included more than others. This is Colorado, Wyoming, but you also see a little bit of Texas down there and kind of the panhandle of Oklahoma. So what probably is most immediately noticeable about this map is all this blue. And this is all outcrop, which is stuff on the surface. So if you've ever been driving down like a highway, for instance, and you've seen kind of these big kind of layered rock formations, that would be outcrop. And a lot of that stuff was deposited, millions of years ago. So what we did was we looked at a lot of these outcrops and a lot of the data that other scientists have done. They've looked at it and they've dated a lot of these areas. So we've taken stuff that existed during that 157 million years ago that was kind of deposited near that time in late Jurassic. And we've plotted it here on this map. And what that allows us to do is it allows us to, in conjunction with looking at the actual like rocks themselves and what type of rocks they are, maybe like sandstone or mudstone, it allows us to recreate what the environment might look like. But of course, we don't only want to look at what's on the surface. So we have some well log data here, which was taken from underground. As you can see, there are all these little dots. The different colors are just sources they came from. So just a way of identifying that. But you'll see how these are kind of in places that we don't get with these outcrops and kind of some of that white space, which is good because that gives us more of an idea of what certain areas of the environment might look like that we might have had to infer before if we didn't have this data. And what we do with those things is we start kind of filling the blanks and we get something like this, which if you see all of this kind of marine area here, this water, you see this coastal kind of orange regions, and then you see down here these Eolian deposits, which would have been like a dune, for instance. So just using the information that we have, we're able to infer what this environment would look like. Now, of course, we don't want to just send this off without kind of double checking it. So the first thing we do that is we look at fossils. So if you come over here, if you see all these kind of red brown dots, these are terrestrial fossils. So they come from animals that would have lived on land. And you can see how they're kind of clustered in these dune areas and kind of on the coast. And then you'll see all these kind of blue ones, these aquatic ones. You'll see that these are all kind of clustered in these marine areas, which is important because if you find, say a marine fossil, typically it's not going to be in a mountain because fish don't live in the mountains usually. So that allows us to kind of confirm some of our observations and confirm how we inferred. And the plan is with all this is to end up sending it off to artists, first fact checkers, but then artists who can interpret this and make it into sort of a satellite image that looks something like this, the one from before, which looks like something that you might see on Google Maps, for instance. And you can kind of see how it gives you a nice visual of what this region could have looked like back then. But what's important about all this is when we're making these paleo maps, because a lot of people have made paleo maps before, but we're being transparent with our data, which is we're kind of outlining how we made these and where the data came from, which for a lot of other ones, it's kind of hard to find that. And that allows two things. For one, it allows there to be kind of a central source of information for this area. But it also means that if a scientist wants to make a map of a different region, they have the resources and the tools to do so, and they know kind of a process on how they can do that. So it serves also as kind of a foundation for a science in the future. So that's how we went about over the summer, trying to recreate what the past might look like using what we have now. Thank you.
Awesome job, Wesley. That's fantastic. That's so cool to kind of get a glimpse into a little bit of the landscape that we all live in currently to see what it looks like. So awesome job. Thank you so much. Awesome. So we are actually going to head down to the bowels of the museum with our entomology and arachnology scholars. And see they're all together over there. Why don't you all take it away? Let us know what's going on.
And yeah, we have it. Okay. Hi, we're the Entomology and Arachnology Teen Science Scholars for this year. I'm Danny.
I'm Sarah. I'm Megan. And I'm Sophia.
And these are our mentors for the season. Frank, Jeff, Paula, And those were elementary. So what is entomology and arachnology? Entomology is the study of insects. So that includes things like butterflies, beetles, flies, bees, and all those sorts of creepy crawlies. And arachnology is the study of arachnids. So spiders, mice, and scorpions, and more.
So as teen science scholars, we're working in both fields, but in general, we have an overarching project. Right now, the museum is in the process of renovating a space called Nearby Nature. This space is looking to be renovated in about the fall, and it's kind of a nature outdoor design plan in which they want to introduce native plants and have it as kind of a play area of natural life. Our job as teen science scholars is to capture and catalog what insects we find to see how this changes with development. This is the second year of a 6-year program, and this is some of the work from last year's teen science scholars, and we are simply continuing on that work with this project.
So what do? So a regular day is Tuesday and Wednesday at 10 a.m. We are live at the museum and we sort the bycatch. And then we spend about an hour catching in City Park at 1 p.m. to 2. And then we come back to the lab, label our catchings, and then work on pinning the insects we caught. And then we leave the museum at 4. And then when we do night catching on Thursdays, we arrive at the museum at 5. We sort on bycatch or work on our pinnings. And then at 7, we go outside and set up the materials. And then we wait until it gets dark and catch. And then at 10, we leave the museum. So this is the entomology lab where we work in. So we have the desks on the left and then the computers. And then this is how to preserve specimens. So first you collect it. So either the bycatch or the actual collectings from City Park. Then we identify them and then we sort them if it is the bycatch, and we sort it into orders, and then we pin it and preserve it.
So going into more detail with that, so when we were sorting bycatch from pitfall traps set by Dr. Fishing, the museum's main arachnologist, which she has collected from various locations around the Southwest. So bycatch is anything that's caught that is not the intended target while collecting. So for us, that contains insects caught in arachnid studies. So we originally get these insects in large jars of alcohol, which are then poured into smaller trays and sorted by order into jars containing labels and 75% alcohol. So common orders we find in these are Hymenoptera, which include bees, ants, hornets, wasps, the like. Diptera, which are flies, Coleoptera, which include beetles, and Lepidoptera, which include butterflies and moths. So this is sort of what sorting looks like. So as you can see on the bottom left, that's what a big tray full of bycatch, and also we get lots of vegetation in them. So that's what that would look like. And it's currently at 75% alcohol, so you can go up to the top right and see the beginning process of sorting it out into orders. And if you go back down into the bottom left, you can see the jars with what looks like much cleaner alcohol. And so that's the 75% alcohol that the individual orders are sorted into. So our other job is collecting specimens for City Park for the Nearby Nature project. So we do that using our soup nets, which we swing in the grass on the bugs fall in, our aspirators, which is basically a straw that we suck the bugs into a jar with a mesh to make sure they don't get into our mouths. However, if you have, and our kill jar, which we put the bugs that we collect in our sleep net and our aspirator into, that has toilet paper with ethyl acetate, which euthanizes the specimens. So this is some images of the field work that we do in City Park, us collecting, and then this is the general area we're collecting. And then finally, storage. Of course, like with bycatch for soft-bodied arthropods, such as spiders or general arachnids, we store them in 75% ethanol alcohol with labels that we've made ourselves. Our main form is pinning, however, for that when the exoskeleton will hold all the info needed when it's dry, we will pin them using various sizes of pin. And for specimens that are too small to pin, we use glue boards, which are pieces of paper stuck on the pins. And so these are some examples of some finished boards we have done. They are categorized by date.
All right, yeah, so this is an example of some of the stuff we have caught in City Park. You can see that we may spread them onto pinning boards or put them in jars. Now, as for what we have actually found in City Park, specifically the stuff we have pinned, we found over 900 arthropods, 70 of which being arachnids. Overall, unsurprisingly, we have found a great number of Diptera, flies, Coleoptera beetles, and Hemiptera, which are true bugs, which might range from like assassin bugs or also leafhoppers. So some of the things we have experienced similar to other groups' field trips are these following experiences.
So we've done things called light trapping, which is what we do on Thursdays. As many arthropods are nocturnal and navigate by light, setting up a bright UV light, insects are attracted and we're able to grab them off of a sheet that displays the light using our aspirators and kill jars.
And then for the science on the spot, we set up 2 tables in the museum main atrium with collections from previous teen science scholar interns and a sample of our collections from City Park this year. We spent the time educating guests about the Nearby Nature Project and answering questions about the insects on display, as well as doing a live pinning demonstration.
So that's what we did in general. But of course, each of us came to this program for different things, and it's important to see what did we gain from this experience.
So I joined this experience because I've always been very interested in animals and I want to continue to have a career in animal related studies. And so that also includes arthropods, which is what we studied here. I was able to really see what a career as an entomologist might look like, which is again, a field that I'm very interested in. One of my favorite moments was light trapping, as it was incredibly fun waiting for the sun to go down and talking about all kinds of stories. And my favorite arthropod changes every 15 minutes. However, I do hold myself, I do own 2 jumping spiders. So those are pretty high up there.
So personally, I want to join this because I hope to go into a STEM field in the future. And I want to get experience working with professionals in a lab-like setting, or an actual lab setting rather, and see what life as a scientist is like. I was able to learn both about the scientific community and what the future in museum science could look like, which I tend to explore now as I think it's very interesting. Night trapping was also one of my favorite moments of teen science scholars of the program. We had many interesting specimens we went up during the day, and also it was good bonding time. And my favorite arthropod is Jerusalem crickets, which, as you can see on the left, which I discovered through this.
So why did I join as an aspiring botanist? I wanted to advance my understanding of the insect world to aid in my studies. I took away that insects are not just a factor of the plant ecosystems, but they have their own unique one. My favorite moment was asking Jeff millions of questions every day, like completely unrelated, and then having very interesting long conversations. And then my favorite arthropod is a dragonfly, which they bite, by the way. Yeah.
All right, I joined because in the future, I'm looking to do a career in wildlife biology or zoology, and this program specifically was to get good experience with ID and fieldwork. What I took away from this is that I was able to talk to a lot of people in a general biologist career path and get their insight for both college and also future employment. My favorite moment was one day before we went night catching, we actually went to the Denver Zoo for an entire day and then came back and night catch. And my favorite arthropod is actually very specifically a white-lined sphinx moth that I was able to catch and pin with the help of one of my other interns. So we just want to thank everyone for listening, and also a big thank you to our mentors.
That's fantastic. Thank you all so much. That was a great presentation. Thank you for sharing a little bit about what you learned and what you took away as well. I know that bugs are not everybody's favorite thing to be around all the time, but I know that the work that you all have been doing is extremely, extremely important. So thank you so much for your hard work and for a great presentation. Awesome. So next, we're going to travel over to the Paleontology department to hear from our scholars over there to see what they were up to this summer. So Paleontology, why don't you take it away?
Hi, so we were the 2022 Vertebrate Paleontology Teen Science Scholars. My name is Lucetto, and
my name is Johannes Mahari.
Okay, so Johannes.
So basically for the first week and a half of the internship, we were basically studying dog skeletons and other vertebrate skeletons, trying to memorize all parts, all their bones. And we were putting wolverine bones together, beaver bones together and stuff like that, just to solidate those bones into our mind. And a couple of reasons why this is really important is because if you're in the field and you see say a hadrosaur femur, you like would be able to recognize it because like all vertebrate bones are basically the same. So like if you were in the field, you would be able to recognize like, oh, this is part of the caudal vertebrae of a hadrosaur. And you can see that the dog's femur looks relatively similar to the hadrosaur's femur on the right, and they're roughly in the same place. So that kind of helped us like know what we were working with downstairs.
So while we were working there, we worked mostly on fossils that came from New Mexico that were collected on BLM land. And they all came from the late Cretaceous and Campanian period. So they were all about 75.2 million years old. And so a lot of the stuff that we got had come straight from the field. And the first thing you have to do is field cataloging, which basically is just unpacking, organizing by date, making sure that they are in clean-ish boxes, and sorting out the ones that need to go to the lab because they were broken or needed to be taken out of jackets, stuff like that. After that, they came down to us and Johannes would catalog them.
Yeah, so basically cataloging would be like, we would give them each distinct EPV numbers so that like that number would travel with them like throughout their whole life in collection so that we could track them down. So we would put them in Excel sheets. We'd put things like where were they found, what specimen is it, what part of the body is it, like what bone, or yeah, like what bone it is and stuff like that. And for big bones, we would put them in boxes and like put tiny little bone labels with the EPV number on them so we could track them. And for small bones and fossils, we would put them in little friction fit boxes. And and for for the really, really small ones, we would put like for a croc tooth about 1/2 centimeter long, we would put those croc teeth inside vials with cotton on them, and we would put those labels on them too.
So there were also some that were like too big or too fragile to go into boxes with foam on the bottom or friction fit boxes. So I ended up making a lot of foam homes, which are just cases made out of foam for those fossils. And so basically what you have to do is you sort them onto the foam and you trace them to make sure they're in the right places. You carve it out and you put Tyvek around the sort of hole so that they aren't damaged while in collections. And this helps them keep them safe. So this was for like irregular sized ones or anything like that. And then after we put them in their boxes or rehomed them, they would end up going into collections. Yeah.
So basically, after we put all their information inside Excel sheets and we put all their labels inside the boxes, that would go into collections. And collections for us is like really organized, and they have like their own separate drawers for each like different families. So like say we got like a lot of Tyrannosaur tooth fragments and a lot of like horns from ceratopsians. So those would go in like those would go in different drawers inside like one side of like a whole cabinet so that they could be well organized.
Yeah. So that was the majority of what we did, but we also helped an artist reconstruct a portrait based off of this skull of a Pentaceratops. And so this was the portrait. And the thing was, a mostly complete Pentaceratops skull had not been found before DMNS found it this summer. So we helped Andre Tuchin, a paleo artist, modify it so it could become more scientifically accurate. So here are some of the changes that were made. Just pointing them out. So stuff like he added this brow bone that was not there before. He changed the shape of the horns on the frills, like right here. The beak is also a lot shallower because before it was way too thick. And that was the other project that we worked a lot on during our time here. And last we did our science on the spot. And so I actually got to show the public the Pentaceratops skull and explain where it was found, what it is, all that stuff, and show off the portraits that we helped change. While Johannes took over some smaller stuff like a hydrosaur jaw that I had put in a foam cradle. I want to talk about that.
So yeah, so basically science on the spot, I was just showing like most of the stuff that was found in northwestern New Mexico. Like there was like a hadrosaur mandible, like the bottom part of its jaw that had a croc bite in it. So that was pretty cool. I got to show the public that, and it was overall a great experience because I got to like physically inspire younger kids to like learn more about what I was working on.
Yeah, it was really nice to see kids come up and get to learn about all the stuff we had on the table and the Triceratops skull, which everyone really loved. And yeah, that was our whole 6 weeks.
Yeah.
Awesome job. Thank you both so much for your presentation and for showing off all that amazing work that you did, even the tedious little labels, getting them on so that they can live on in collections, and we continue to learn from the work that you both did this summer. So thank you so much for your presentation and for sharing with us, to you both. All right, so we are actually now going to head over to our paleobotany scholars to learn a little bit about what they had accomplished and done this summer. So Milo and Mary, take it away.
Hello, welcome to our Paleobotany Showcase. I'm Milo Illustre, I'm 16 years old and I go to Denver School of the Arts.
I'm Mary Ward, I'm also 16 and I go to Thornton High School.
So a little bit about what we've been doing this summer is we have been helping our mentor Gussie McCracken with her research project relating to the oldest beans in the entire world.
And these beans are 65.5 million years old, so they are pretty old.
So we've been collecting data through fossils, and we have been photographing these fossils for her to put them in her paper. So to start us off with our process of being in the field to curation is field work. And here we are in Corral Bluffs, which is where the oldest beans in the world were found. And we are at a specific locality called Baby Backpack that has the second oldest beans in the entire world.
And this is in Colorado Springs, so it's not super far away from us either.
Here is some of the fossils that we found. Here is a palm seed. Here's me digging up fossils. Here's a big pile of fossils. Here's us trekking around looking for spots to dig.
Yeah, and so this is part of the same locality, it's just in a different section of this. Over here, the rock was much more crumbly, it's very steep, and there was lots of sand, so for this process, we used a hammer to break open the rocks at the seam, and from there we would examine the rocks, look for any leaflets or bean pods that we could find. And if we thought we found something but we weren't sure, we would bring it to Gussie and get her confirmation.
And here's the whole team down at Corral Bluffs. And the second step to our process is unwrapping fossils. So this was actually the first thing that we did when we came to the museum. So we unwrapped a whole bunch of fossils that Gussie had collected in that year or previous years. And here is a little bit of the process of how we did that.
So we start off this pretty short process by sorting the fossils by locality. So whenever they are transported from the field to the museum, they're often super mixed up. So we have to sort the specimens by locality. And locality is just a fancy word for the association of the data for these fossils. So this includes things like GPS, location, elevation, the type of rock. From there, we unwrap them and send them to the prep lab.
Here we are collecting data and cataloging. Here is Mary gluing on the specimen number onto one of our fossils. Here is a whole bunch of fossils that we cataloged this summer. Here is me putting away some of the fossils that we cataloged back into the collections. Here is another look at those fossils.
And here we are photographing specimens. This is a pretty long process, but it's really exciting with all of the details. It was very intricate. So here in the top left corner, we have Milo working on a program in which we find the highest and lowest vocality points in what's called stack photography, which Milo will explain in a second. And so you can see we have a pretty nice camera to work with, and it adjusts either on a large scale or a much smaller scale based on our needs. We have lots of different lighting, so we're able to adjust that. And of course, we have to include the scale. Otherwise, all of this is pretty pointless. And over on the top right, we can see Milo rendering these pictures, and that leads us to stack photography.
Perfect. So here is stack shot photography. This was one of my personal favorite parts of this internship, is taking a bunch of photos of these specimens. So here we have a super in focus photo of this bean, but this wouldn't look like this if it weren't for stack shot photography, because the way that stack shot photography works is that we measure the lowest and highest points of focus, and this camera will take 20 to 60 or 70 photos of each point of focus in between the highest and the lowest point. And this is significant because often on these plant fossils, the surface is not smooth, so it's hard to have the whole entire fossil in focus, which is what we needed for these photos. And here we have a leaflet and a bean pod, and as you can see, these are super in focus. And after all those photos are taken, they are put in another program where they are all stitched together to create this nice photo. Here's another one of a leaflet.
And here we are in our field trips. So on one of our field trips, we got to join Anthropology for this.
Here's us reconstructing a bison skeleton. This bison had passed away about 3 years prior to us reconstructing it. And this was a little difficult because we didn't have like instructions to complete it. And here is the deer that we saw at Rocky Mountain Arsenal. And here is a bison named Ferdinand, and he really loved his flower patch. All right. Thank you so much.
Yeah, thank you.
Yep, I really enjoyed this internship, and it was one of the best experiences of my whole life.
It was absolutely amazing and would do it again.
For sure. All right. Bye.
Awesome job to you both, Mary, Milo. That was fantastic. Thank you for sharing all the work that you did in our paleobotany department this year. So that's it, everybody. Those are all the areas. So let's give a round of applause, virtual round of applause for all of our teen science scholars and their presentations this year. You all did fantastic work. And now, if any of our participants, any of our attendees, we have some time. If any of you do have any specific questions that you have for our scholars, we'll carve out a little bit of time. You can use the chat function, or you can use that Q&A button down below there to ask questions, and then we can ask them out to our teen science scholars here. But again, while waiting for those to come in, I want to thank again so much. Like I said, this program takes a village to be able to run year after year with it. I know it's only 6 weeks, but there's a lot of hard work that goes into it. So again, I want to thank the TSS committee who works with me in this program. I want to thank all of the mentors. You guys do so much amazing work with our teens. So thank you, thank you. I want to thank again the friends and family who support our teen scholars every year coming in, especially this year. Thank you for supporting your scholars to participate in the program. Thank you again to our contributors and to our donors for this program. This truly could not run without your generosity. Thank you so much for supporting this program in the way that you do. It means the world to us as an institute. So thank you. And of course, one final humongous thank you and congratulations to all of our teen science scholars this year. All 20 of you have been immensely impressive from the very first moment that we got to meet you till right now and the whole way through. You have all been so fantastic to work with. Can't wait to celebrate with you all next week of your accomplishments, but thank you for your contributions. You all are fantastic. So thank you, thank you. I will check real quick. Doesn't look like we have any questions because your presentations were all so thorough and amazing. So way to go. So we will actually end our showcase presentation with a little bit of time to spare. Oh, it looks like, oh, we do have one question for the anthropology department. It's asking for a little bit more of an in-depth explanation of flotation. So can one of or a couple of our anthropology scholars go ahead and unmute yourself? And can you talk a little bit about what flotation is in a little bit more detail?
Sure. I'm sure the actual audio itself on the video was probably a bit laggy, so you probably couldn't hear. So flotation is basically a process in which you use water and you use a bunch of dirt as well. So we have our samples in one area, and so we dump our soil samples into the dirt, and then we allow all of the light matter and organic material light matter to rise to the surface. And then we pour that into like a cheesecloth. And so that would be stuff like charcoal, pollen, twigs, stuff like that. Charcoal, pollen, seeds, that kind of thing. And so after we would drain out that water, there would be a bunch of heavy matter at the bottom. So that would be including things like bones, pottery sherds, spear points, stuff like that. And so we would also put that out onto a newspaper for it to dry, and we would sometimes screen it as well if the sample was really, really important. And so that is usually called a heavy fraction, and we tag everything during that process. So that's basically what flotation is, and that's basically what we did. I hope that answered your question.
Awesome. Thank you, Faria. Okay, any other last questions? Awesome. Well, again, thank you all so much for carving out some time to hear all these amazing presentations. I hope that you all have a fantastic rest of your weekend. And thank you, thank you, thank you. We'll see you all soon. Bye, everyone.
Bye.